Okay, let's do the write down. We did the C3 photosynthesis. Can you rewrite the entire Calvin cycle? So I'm going to add the arrows. You have to write down whatever the things that are coming into the cycle and then the outputs. Just complete this. Okay, complete this, huh? So we are bringing carbon dioxide into the Calvin cycle with the help of the RUBP. We're going to fix 6 carbon dioxides. And for this, we are using the Rubisco as the enzyme. And we are making 12 PGA. I need you to remember the numbers, huh? 12 PGA phosphoglyceric acids. And 12 PGA going to reduce into 12 G3P. From this 12 G3P, we are going to use 2 of them to produce glucose. I forgot one box. You have to have glucose. So glucose is the substrate for cellular respiration to produce more ATP. And I'm using 10 G3P to regenerate 6 RUBPs. So we can do the Calvin cycle again and again and again. But Calvin cycle is going to use a lot of energy. It's going to consume ATP. And it's going to consume NADPH. Right, so this is what you learned last week. If you want, you can break it down and put the whatever the ADP and PI and NADP and H+. Right, so this is the full complete C3 photosynthesis. So the first stable product in C3 plant is the 3 carbon PGA. Draw the carbons as well. RUBP has 5 carbons. carbon dioxide bringing 1 carbon into the Calvin cycle. Split it into 2. PGA is a 3 carbon compound. G3P also 3 carbon. There are no past exams. No? I'm going to do every single possible question. This is like brand new to the study design. This is the first time students, I'm teaching this. Right? There's no past exams. So this photosynthesis is the normal photosynthesis. So this photosynthesis is the C3 because the first stable compound is the PGA. It's a 3 carbon compound. Okay, write down the temperature. You can find the C3 photosynthesis. You can see the C3 plants around 25 to 35 degrees, right? Like the normal environmental plants. So when it comes to the C3 plants, they always live in good environmental conditions. Like their temperature is low and the humidity is high. So because of this, they will always do, most of the time, they will undergo the normal Calvin cycle. Right? So the C3 plants, they always do the normal Calvin cycle. But sometimes if the temperature increases, decrease in the humidity, I did all these things last week. Sometimes if the temperature increase beyond 35 degrees, plus the humidity needs to drop, that means it needs to be very, very dry condition. Then the C3 plants need to shut their, close their stomata. Then they will undergo the photorespiration. Right? I don't have space. I'm going to go to the next page. So rewrite this one. C3 plants are the ones around like the normal temperatures. Most of the time around this range. So they will experience the lower temperatures and higher humidity. Right? So if they experience this condition, they can leave the stomata open. So if they leave their stomatas open, they can get the carbon dioxide in. So they will do the normal Calvin cycle. Right? So most of the times the C3 plants can leave their stomatas open. That means they will undergo Calvin cycle, producing glucose to produce ATP from the cellular respiration. But sometimes if the temperature goes beyond 35 degrees, decrease in the humidity, then they can't, C3 plants can't leave their stomatas open, so they're going to close their stomatas. If in case, if they need to shut their stomatas, they don't have carbon dioxide for the normal Calvin cycle, so they're going to switch to the photorespiration. Right, so this is what I did last week. So remember, C3 plants most of the time will undergo the normal Calvin cycle, right? So if something happens, like if the environmental temperatures increase, decrease in the humidity, then they will undergo photorespiration, right? So this is something happen like sometimes. So because of that, C3 plants won't have any adaptation to avoid photorespiration. right? So the only plant that you're going to see the photorespiration are the C3 plants, because this is not something they're always going to experience. If this happens sometimes, they can't avoid the photorespiration. They don't have any adaptation to avoid the photorespiration, right? These are the questions. Write down this. C3 plants don't have any adaptations. I'm going to do C4 and CAM. You will see adaptations to avoid photorespiration. But C3 plants most of the time experience the good environmental conditions. Sometimes they will undergo the photorespiration, so they can't avoid the photorespiration. But if plants like C4 and CAM, I will explain what the CAM is. C4 CAM are the plants that live in very hot and dry conditions permanently forever. So they must have adaptations to avoid photorespiration, right? So C3 don't have any adaptation, but C4 CAM, hot environmental, like the desert plants, will have adaptations. Right, I'm going to start with C4 and then we're going to do the CAM at the end. Right, so the C3 plants are C3 because the first stable product, PGA, had 3 carbons. So let's do the C4. Right, all these things I did last week. Huh? So I'm going to go to the C4 photosynthesis. For C3, I said the plants live in around 25 to 35 degrees. These are rough estimates. Huh? So now I'm going to increase the temperature. C4 means something like around 35 degrees to 45 degrees, right? So it's higher temperatures than the normal C3 plants. Okay, I'm going to give some examples of C4 plants. So if you think about all the available plants, most of the plants are C3, right? I don't know the percentage. Let's say like 90% of the plants, don't write this down, but lots and lots of plants are C3 because they live in like good environmental conditions, right? But you can just, the rough estimate of the C4 is around 3%. This is correct, right? I don't know this one. So some desert plants live in like hot environments will undergo C4 photosynthesis. Write down the examples. Most of the grass species, most of the grass. I want you to remember these 3 examples. Huh? Sugarcane and corn. You might get examples. I can give 4 choices with asking you to pick the correct C4 photosynthesis plant. Right, these are common examples. So go to the sugarcane, corn, and the grass. Any grass species. Most of the grass is going to be C4. Right, okay, so C4 plants live in hot environments, right? So they're going to be in high temperatures and dry conditions. So they must have adaptations to avoid C4. They must have adaptations to avoid photorespiration, right? I will go through C4 adaptations at the end to avoid photorespiration. So let's go through how the C4 plants going to produce their glucose. So they're going to first trap the carbon dioxide. So inside their cells, they will do the carbon fixation first. So if a plant is going to produce glucose, the first thing they must do is they must trap the carbon dioxide, right? Because carbon dioxide is the one that gives the carbon for the glucose. So first step, C4 or C3 or CAM is fixing the atmospheric carbon dioxide. But for this initial fixation, C4 plant's going to use something else, right? So I'm going to come back to this. Can you write down this somewhere else? Complete this, huh? So if I use the RUBP, the 5-carbon RUBP, plus carbon dioxide, write down the enzyme we are using. So this is the carbon fixation done by the C3 plants. They're using a 5-carbon ribulose biphosphate, RUBP, to trap 1 carbon dioxide to make PGA. Right, so the enzyme is Rubisco. So without writing the enzyme Rubisco, I can write down instead RUBP carboxylase. RUBP carboxylase is nothing else, it's Rubisco, right? So another way to write down Rubisco is you can write it down, it's RUBP carboxylase. Right, so without writing Rubisco, I'm going to introduce something. So without writing Rubisco, instead I'm going to use the term RUBP carboxylase. Can you highlight the RUBP part? So the Rubisco or the RUBP carboxylase, the initial substrate is the RUBP. Right, so RUBP, the 5-carbon RUBP, going to trap a carbon dioxide to produce the final product, PGA. But the disadvantage of the Rubisco is it can either bind with carbon dioxide or oxygen, right? So it doesn't have to be for Rubisco always carbon dioxide. It has the same affinity towards the oxygen as well. So can you write down the oxygen example? So here it's binding with the oxygen, not with the carbon dioxide. Right, there's no PGA, there's no Calvin cycle, you're going to stop it, right? So this is the photorespiration. So this is what happened in the normal Calvin cycle. The initial carbon fixation done by the Rubisco enzyme or RUBP carboxylase, but the substrate here is the RUBP, right? But Rubisco is not a good enzyme because Rubisco or RUBP carboxylase can either bind with carbon dioxide or oxygen. Can you write down underneath? Rubisco is a bad enzyme, right? Because if in case it binds with the oxygen, there's no Calvin cycle, no glucose production. Instead, it's just going to undergo something called photorespiration. Right? Photorespiration won't produce glucose. right? So the one of the examples, one of the adaptations that are going to be in the C4 is they're not going to use the Rubisco for their initial carbon dioxide fixation. Because if they use the Rubisco, most of the time they might bind with the oxygen, not with the carbon dioxide, right? So they are always, the C4 plants are always in hot environments. They have to adapt to avoid the photorespiration. But they can't avoid the photorespiration if they use the Rubisco for this initial carbon dioxide fixation. So what the C4 plant is going to do is, C4 plants for the initial carbon dioxide fixation, they're going to use something else. So I'm going to write it down. Just write this down, then when I rewrite it on the other page, you will understand this. Huh? They're not using, C4 plants not using the RUBP carboxylase or Rubisco for the initial carbon dioxide fixation. They're using something else. I'm going to write down the enzyme name. The enzyme name is PEP carboxylase. Can you write down the substrate they need? If the C3 plants use RUBP carboxylase, the initial substrate is RUBP. If the C4 plants use another enzyme, PEP carboxylase, the substrate they need is, write it down, PEP. Right, so if you highlight the PEP part, it's easy. Okay, write down PEP. RUBP has 5 carbons, PEP has 3 carbons. Can you count the carbons? So this is the C4 plants, right? I will rewrite everything, you will understand this. But if I say the substrate is 3 carbon PEP, it's bringing 1 carbon dioxide. Write down how many carbons in the product. Right, 3 plus 1 is 4, right? So can you write it down? We're going to form a product having 4 carbons. PEP is a 3 carbon. PEP is going to bring 1 carbon dioxide from the outside, and it's going to form a product which is going to have 4 carbons. So this product's name is, write it down, there's only like few substrates in any ones, huh? And then it's going to go through the normal Calvin cycle again. Write down the first stable product the C4 plant is going to form is the oxaloacetate. Oxaloacetate is a 4 carbon compound. And can you write down underneath, PEP carboxylase is more efficient because it only binds with carbon dioxide, never ever with oxygen. So C4 plants, the initial carbon dioxide fixation is by an enzyme called PEP carboxylase. Right, so PEP carboxylase is more efficient because it can only bind with carbon dioxide, never ever with oxygen. They're not going to do anything called like the photorespiration. But this is a different reaction to the previous carbon fixation. So if you use the PEP carboxylase, we need PEP as the substrate, a 3-carbon compound, and it's going to bind with 1 carbon dioxide from the outside. So the product going to have 4 carbons. So the first stable product the C4 plant is going to produce is the 4-carbon oxaloacetate. You have to know every single thing on this slide. I'm going to rewrite the C4 again. Huh? We're going to rewrite everything like a couple of times until you get used to these things, inputs and outputs. Everyone finish this? Can I go back? Done, yeah? I'm rewriting this part now. I just introduced the PEP carboxylase. Can you go back and write down? So I'm going to rewrite the carbon fixation. So write down number 1 is we're going to do a carbon fixation. So rewrite whatever we wrote. So the enzyme we are using is the PEP carboxylase. So the substrate needs to be PEP, a 3-carbon compound. It's going to bring 1 carbon dioxide into the cycle. So the first stable product, 3 + 1, needs to have 4 carbons. Name is oxaloacetate. Right? So you have to remember PEP carboxylase and oxaloacetate. Can you highlight the oxaloacetate? So oxaloacetate is a 4-carbon compound. And this is the first stable product produced in C4 photosynthesis. That's why the plants are C4, because the first stable product produced during this C4 photosynthesis is the 4-carbon oxaloacetate. It has 4 carbons, so the plants are C4. These are the possible questions. Huh? So you have to know the enzyme name and you have to know the first stable product, oxaloacetate. Right? So one more new term and then it's the same thing again and again. Everyone understand up to the oxaloacetate, right? Okay, write down oxaloacetate is going to convert into one more final product. That's it. No more new things, right? So can you write down oxaloacetate going to break down into another compound? You can just go across. The last one is the malate. Some people might call it as the malic acid, the same thing. Huh? Malate or the malic acid. So oxaloacetate will form malate or malic acid, and it's again 4-carbon compound. That's it. No more new things. This is it. Right. So this is the first initial carbon fixation inside the mesophyll cells. So if you remember what we did last week, mesophyll cells means it's around these cells like closer to the stomata, right? So they can interfere with always the carbon dioxide or oxygen. So always the initial carbon fixation is in the mesophyll cells. So C4 plants, they're going to have a special adaptation. For the first carbon fixation, they're using a special enzyme, not the Rubisco. They're using PEP carboxylase. So their substrate is PEP, a 3-carbon compound, bind with the atmospheric carbon dioxide to produce 4-carbon oxaloacetate. Oxaloacetate is the first stable product. A C4 compound will break down into malic acid. That's it. So this is the first initial carbon fixation. Are there any questions? I'm going to do lots and lots of questions at the end. You need to remember these things, right, to do the questions. So if you understand this, let's go to the bundle sheath cells. So I did the plant structure last week. You have to know where the mesophyll and where the bundle sheath cells are, right? So bundle sheath means something around the vascular bundle, right? So these are the bundle sheath cells. So we are going to move the malic acid or the malate into this bundle sheath cells now. So let's go to the bundle sheath cells. So I'm going to bring the malate or the malic acid into the bundle sheath cells. I'm going to use the term malate, but in some books they use the malic acid, right? Whatever, the same thing. So can you break down malate into a 3-carbon compound? I don't think you need to know the name of this. Just leave it as 3-carbon because I'm going to remove 1 carbon as carbon dioxide. Malate is a 4-carbon compound. It's going to release 1 carbon dioxide inside the bundle sheath cells. So the bundle sheath cell is going to have carbon dioxide to undergo normal Calvin cycle. Can you rewrite the normal Calvin cycle again? So the C4 plants must undergo Calvin cycle because they must produce glucose. But they're going to have 2 carbon fixations. One is to fix the atmospheric carbon dioxide using PEP carboxylase. And they're using carbon dioxide, they're making a 4-carbon malate. And they're going to transport the malate into the bundle sheath cells and release carbon dioxide again. Right, so this is the carbon dioxide coming to the Rubisco to undergo normal Calvin cycle. Can you rewrite the Calvin cycle here? I'm going to redraw it. I'm going to get space. Draw it up. So when you write like Calvin cycle lots and lots of times, you will get used to the Calvin cycle. I'm going to bring the malate in. So you can call it this as the Calvin cycle. Right? So this happens inside bundle sheath cells. So transport the malate into the bundle sheath cells and break it down to get the carbon dioxide again. But this is not the atmospheric carbon dioxide. This is the carbon dioxide coming from malate. Right? The Rubisco not seeing any oxygen. It's only going to see carbon dioxide. So write down exactly the same thing. We are using RUBP 6, 6, 12 PGA, 12 G3P, 10 G3P. I'm writing exactly the same thing. Write down Rubisco. C4 use Rubisco again. Huh? But they're not using Rubisco in the mesophyll, they're using somewhere else, away from oxygen. Rubisco is only seeing carbon dioxide, it's not going to be see any oxygen. You don't have to know this 3-carbon compound, but malate 4 carbon, remove 1 carbon, so the product is 3 carbon. I posted the notes, everything in the note as well. Are there any questions? Neha, I can't see you. You there? Neha, come on. Is this clear? So the only difference is this: the C4 will undergo normal Calvin cycle, right? But they're going to have, there's a 2 carbon fixations. So they're not using Rubisco for the initial carbon fixation, they're using more efficient PEP carboxylase, right? So PEP carboxylase is more efficient because it can only bind with carbon dioxide, not with oxygen, right? So they're going to completely avoid the photorespiration. So PEP carboxylase disrupts carbon dioxide and it's going to produce oxaloacetate and convert into malate. And this is the malate, the compound, which is going to give the carbon dioxide for the Rubisco enzyme. Rubisco is not dealing with the atmospheric carbon dioxide. It is getting oxygen, it's getting carbon dioxide from another compound, right? So the problem comes if the Rubisco try to bind with something, the atmospheric gases, right? So because it's a combination of both oxygen and carbon dioxide. But if you keep the Rubisco somewhere away from oxygen, giving only the carbon dioxide, Rubisco will only undergo Calvin cycle, producing glucose for the cellular respiration. Right, so I did the C4 photosynthesis. The first stable product is the oxaloacetate. That's why the plants are C4. And C4 plants going to have 2 carbon fixations. I'm going to compare every single thing at the end. You will understand what I'm saying, right? Are there any questions? I'm just repeating myself. Okay, write down 3 adaptations that shown by this C4 plants. So C4 plants going to show adaptations because they're permanently living in hot environments, right? So they must have adaptations to avoid photorespiration. Otherwise, no Calvin cycle, they're going to die. Not like the C3, right? They're permanently living in bad conditions. So they have completely avoid the photorespiration because they have adaptation. So can you write down 3 adaptations, C4 plants adaptations to avoid photorespiration? So can you write about talk about the locations? And can you write about the the 2 carbon fixations. The first one, they're using a special enzyme. And the third one is they're having the Calvin cycle, but this enzyme not getting the carbon dioxide from the outside, it's getting from somewhere else. Right? Rubisco have the problems. Rubisco is going to have problems if it is going to try and trap the atmospheric carbon dioxide. I'm giving you clues. These are not the points, right? Write on the points. Right? Okay, I wrote them down. Just check whether you got this. So C4 plants, not like the C3, they're going to have 2 carbon fixations. The first initial carbon fixation is in the mesophyll cells. And then the normal C3 Calvin cycle going to be somewhere else, right? So towards inside. Can you remember the Kranz anatomy I did last week? Right? It's something special only in the C4 plants. So they can separate the basophils and the bundle sheet cells, right? So they can separate the initial carbon fixation from the normal Calvin cycle. C4 plants use a special enzyme for the initial carbon fixation, which never ever binds to go and bind with oxygen. It's only have affinity towards the carbon dioxide. C4 plants going to keep the Rubisco enzyme inside the bundle sheet cells and give carbon dioxide from another compound. They're not getting carbon dioxide from the atmosphere, right? So it's going to have a steady supply of carbon dioxide. Rubisco is going to have a constant carbon dioxide supply, right? Rubisco not going to deal with the oxygen because it is going to be kept inside the bundle sheet cells, away from oxygen. So you can write down the C4 is more efficient. Huh? So if you compare the photosynthesis rates between C3 and C4, C4 is more efficient because Rubisco is getting carbon dioxide not from the atmosphere, it's getting the carbon dioxide from Malay, right? The same exact reason. So if you get carbon dioxide from another compound, it's going to give a constant supply of carbon dioxide. It's always going to be present. But the C3, the Rubisco needs to rely on the outside environmental factors. But the C4 is getting, the Rubisco enzyme get the carbon dioxide from another compound. So it's going to have a steady input of carbon dioxide. So it can produce more glucose than the C3. C4 is more efficient. Is this clear? Are there any questions before I do this CAM plants? CAM is exactly the same as C4. How did you finish? Yeah. Okay, I'm going to do the CAM plants, right? So we did the C3 plants. Can you write down the temperature? I did the C4 plants. Can you write down the temperature? So what is the temperature C3 plants can live in? C4. And the last one is the CAM plants. Right? So the normal warm temperature is C3, but a little bit hot. C4. I said grass, sweet corn, not normal corn, and then sugar cane. And beyond 45 degrees, right? More than 45 degrees is the CAM plants, right? So let's do the normally like the desert plants, right? So CAM photosynthesis. So this is a plant species, right? They got the name from a plant species, not because of a stable compound like these two, right? So it's in the note, you can read it. So write it down, you're doing CAM photosynthesis. Right, so greater than 45 degrees. Can you write down a couple of example plants? Right, the first plant, because the plants are living in very hot environments, the first thing is they must have adaptations to avoid the photorespiration, right? So this is extremely hot conditions above 45 degrees. So they have adaptation. Most of the CAM plants can store water in their leaves. Right, so this is the clue. Now think about the plants that are going to have like the water stored in their leaves. So you might have these plants in your garden. Right, the first one is the cactus, right? The cacti always. one example, another example is the pineapple. So this is like a pineapple is a common example. Write it down. Pineapple is a CAM photosynthesis. And think about like a horticulture plant which stores lots of water in the leaves is the succulents. So if I ask for CAM plants photosynthesis adaptations, you can see they store water in their leaves, right? Because they are experiencing extreme transpiration or water evaporation. So they must store their excess water somewhere. So they're going to store water as much as they could inside the leaves. Right, so the CAM photosynthesis is exactly the same as the C4 photosynthesis. So can you write it down? The initial carbon fixation and then the Calvin cycle. Exactly the same thing, rewrite it. Rewrite it like a couple of times, then you will use to these terms. Don't write the mesophila and the bundle sheet. Huh? They're not going to be spatially separated. Don't write the location, right? I will do it. But do the other things. You can add the energy cost as well, right? So the previous one, I think I forgot. It's a normal Calvin cycle, that's C4. It's going to use ATP like the normal one. Exactly the same a normal Calvin cycle. Finish. Everyone finish? I can't see most of you. Finish or not? Shall I finish? Finish, right? Okay, so can you write down, go back to the C4, this is exactly the same thing, there's no difference, there is a difference. So go back to the C4. So the only difference is C4 plants photosynthesis. Where is it? So this is the C4 photosynthesis, right? So can you highlight the spatial separation? So the spatial separation means the carbon fixation happens inside the mesophyll, and the Calvin cycle going to happen somewhere else, the bundle sheet cells, right? So we're going to call this as the spatial separation. They're spatially separated two different locations. Right? So if I use the term spatially separated, that means the first part is going to be in mesophyll, and there's going to be the Calvin cycle in somewhere else, the bundle sheet cells, right? So C4's photosynthesis is spatially separated. It's going to take place inside the mesophyll and bundle sheet cells. But if I go to the CAM, this is C4, right? But if I go back to the CAM photosynthesis, this is what I was doing, CAM photosynthesis. Because of the extreme heat, this is more than 45 degrees, right? So we're looking at the temperature more than 45 degrees. So during the daytime, they can't keep their stomata open, right? So they must close their stomata completely during the daytime. So that means they have to fix their carbon during the nighttime, right? So the initial carbon dioxide fixation, carbon fixation is going to happen during the nighttime. But the same location, mesophyll cells. Write down why it needs to be nighttime, not the daytime. Because of the extreme heat, they can't leave the stomata open, right? If the stomata are closed, the plants can't get the atmospheric carbon dioxide. But during the night time, the temperature is going to cool down, so they can leave the stomata open. If the stomata is open, then they can get the gases in and out, right? So the CAM photosynthesis, the initial carbon dioxide fixation must happen during the nighttime when their stomata are open. So once they produce the last malate, they're going to store this in their vacuole. Remember the vacuole? When you draw the plant cell, we draw like a huge vacuole, right? Vacuole's function is to store substances. So one type of thing that's going to be stored is the malate produced during the nighttime. So can you write down the CAM photosynthesis? The CAM plant's going to store the malate in their vacuole, and during the daytime, they're going to transport it into the mesophyll. They will use the malate during the daytime, right? There's no transportation, it's the same place, huh? Calvin cycle also mesophyll cells. Can you write it down? But it's going to be during the daytime. There's no spatial separation. exactly the same spot, mesophyll. We are not using bundle sheath here. So the CAM photosynthesis, the carbon fixation during the nighttime while the stomata are opened, and they're going to store the malate until the sun comes up. So once they have the sun, they will use the malate to get the carbon dioxide to undergo normal Krebs cycle. Right, so this is known as the temporal separation. Write it down somewhere else. So initial carbon dioxide nighttime, Krebs cycle daytime, it's temporal separation. Right, so C4 CAM photosynthesis, the two differences are C4, you can see the spatial separation. CAM, you can see the temporal separation. That's it. I think the questions will use these terms. You have to know this, right? I'm going to now compare stuff, do the comparisons. Huh? These comparisons not in the notes, so write this down properly. This is finished. Shall we do this comparison? Justin, finish. Let's go. Okay, I'm going to compare the Rubisco and PEP carboxylase. Right, so the two photosynthetic enzymes. We did a separate chapter on enzymes. You have to relate this to the photosynthesis enzymes, right? There's no separate unit, separate chapter. So these are the two enzymes, PEP carboxylase versus Rubisco. So if you want, Rubisco is also known as RUBP carboxylase. Compare this. Okay, write it down for both enzymes. Finish? Haven't finished? Okay, let's go through this. So PP carboxylase never ever binds with oxygen, right? Rubisco can bind with oxygen. So you can relate it to the photorespiration as well. Binding with oxygen means can undergo photorespiration. So binds with carbon dioxide at low carbon dioxide concentrations. That is PP carboxylase, because it only binds whether the carbon dioxide high or low, it doesn't matter. It only binds with the carbon dioxide, PP carboxylase. But Rubisco, to bind with carbon dioxide needs to be high, right? High concentration. If the carbon dioxide is low, it can't bind with the low carbon dioxide concentrations. Is this clear? These are not in the notes. So write this down. Okay, next one. I'm going to compare C3 versus C4, and then we're going to compare C3, C4, and CAM. Is this done? Taslim, are you done? Finished? Everyone done, right? Okay. Let's do C3 versus C4. This carbon dioxide acceptor means is it RUBP or PP? I'll give you the option, write down the correct one. Huh? Don't write these. So carbon dioxide fixing enzyme, just make it initial one because there's two for the C4. So what is the initial carbon dioxide fixing enzyme? What is the first stable product of this entire photosynthesis? I wrote all the options. No, I haven't. Finish. Okay, I'm done. Just check. So the first one is C3, the initial carbon fixation. Carbon dioxide accept is the RUBP, 5-carbon compound. C4 is the PEP, C3 compound. There's 2 carbon fixations. So the initial carbon fixation, Ruby score, there's only one, always a C3 Ruby score. C4 is the initial one, carboxylase, PP. First stable compound is the C3 is the PGA, a 3-carbon. Sometimes in multiple choices, I see like 4-carbon, they just give both of them. So if you don't have the oxaloacetate as the option, go for the malate, right? Because both of them are 4-carbon. Sometimes they say this is because of malate, because it's the second stable compound. If you don't have the option oxaloacetate in a multiple choice, go for the malate. Both of them are 4-carbon. So the C4 means 4-carbon. C3, carbon dioxide fixation once, C4 twice. Are there any questions? Okay, next one. Let's do C3, C4 CAM comparison. Anyone writing this down too? Should I finish? Cameron finish? Everyone finish, yeah? Okay, C3, C4 and CAM. List them down. Now I might need to go to the next page, so list them down. I have not. So what is the primary carbon dioxide acceptor, RUBP or PEP? Let's do the initial carbon dioxide fixing enzyme. Right, you have 2 options, PEP carboxylase or Rubisco. What is the first stable product? PGA or oxaloacetate. So this one is new. So I'm going to write down the Calvin cycle. Where the Calvin cycle is going to take place? Number 4. Is it in the mesophyll or bundle sheath? So if you remember the Cran's anatomy I did last week, which one has the Cran's anatomy and which two are not going to have the Cran's anatomy? Okay, I used the same page. What is the optimum temperature? The last one. The temperature range I gave you? Write it down. Normal plan C3. Right, I finished. I just wrote the examples as well. So this is the summary of everything, right? So C3 is the normal plant, C4 grass, sugarcane, corn, cactus, succulent, and the pineapple. Can you write down somewhere cam stores water in the leaves? So it's complete. So this is the next big topic, right? If you have finished the first act by now, this is the photosynthesis, cellular respiration are the next parts. There's lots and lots of content. Start studying, no? Finish? I'm going to send something. Can you start, those who finish, start doing this? If you don't get this, let me know, I'll email. It says leaf structure, a PDF. And do the worksheet. I have another one. Huh? I have lots. Is anyone who needs this? No one? Okay, can you please do this? I sent this document, right? Everyone got this? If you don't have this, let me know, I will email. Don't look at this table. Can you write down whatever the ones we went through last week? Don't get anything from this, huh? No need. Five minutes, hurry up and do it. Okay, let's go through the answers. Is this done? Finished? Finished, right? Okay, first one. So the first box is for the upper cuticle. And then comes the upper epidermis. Second box is upper epidermis. And the third box is the mesophyll cells. The fourth one, I think they are pointing at the chloroplast. Next one is the spongy mesophyll. So this box, they're looking at, I think, the vascular bundle, or you can say xylem or phloem, right? Or this the vascular bundle is enough. Or if you pick it from here, it's vein, right? I did not teach anything vein. Called vein, so just go for vascular bundle or xylem or phloem. So this one here is the lower epidermis. This is the stoma. One single space is the stoma. Stomata is the plural. And then this is the last one is the guard cell, right? I highly recommend, I'm just going through the guard cells. Just read it about this guard cell a little bit. Right? You need to know the stomata, so why not the guard cells? Right, so the last one is the guard cells. Part 2, the movement of water in the leaf is the transpiration A, letter A. The movement of oxygen and water vapor in the leaf is the B, going towards the mesophyll cells. B. The movement of carbon dioxide in the leaf is C, going towards the mesophyll cells. Right, so A, B, and C. What is the name of the structure which carry the water is the xylem. What is the name of the structure which carry the food or the sucrose is the phloem. Right, so this is from last week. I'm going to give lots of multiple choice now. You have 15 minutes. In 15 minutes, I'm going to go through the answers. I'm going to send this. If you need the PDF, I'll email. I'm sending the PDF anyway. Okay, send. So it needs to look like this. If you don't have it, let me know. I will email. do all the multiple choice. I'm giving you until 7:50. There's only 31 multiple choice. Hurry up and do it. I'm going to send a concept map as well. Just save it. Huh? I'm going to give the question and the answer so you can save it. This covers everything, so download this. Concept map. Answer. The concept map. The question. Finish? For those who finish, I'm going to send 3 short answers. Huh? If you finish, start doing this. C3 can short answers, no answers. I'll send the last one, C3, C4 can short answers, right? Answer those questions if you don't. I'm going to go through the multiple choice, huh? Everyone ready? Harini, finish? Sam, you finish? Okay, question number 1. What supplies the electrons to the Calvin cycle? It is NADPH, right? So it needs to be a coenzyme dropping the high energy protons or electrons into the electron transport chain. So NADPH is the answer. So question number 2, light reactions occur in the thylakoid membrane and stroma. Stroma is the liquid, right? So the 2D. 3, stomata are the sites of gaseous exchange. So answer is C. 4. Carbon dioxide is used in the Calvin cycle. Answer is B. 5. Which of the following is not a product of light reaction? We are not producing water, it's an input, right? So answer is 5, D. Next one, which one of the following is not a product of Calvin cycle? It's carbon dioxide. We're using carbon dioxide, not an output. 7 is photosynthesis catabolic or anabolic. It is anabolic because you're building up glucose. So 7 is B, 7B. Which of the following is not a photosynthetic pigment found in plants? We went through every single thing except chlorophyll C, right? So chlorophyll C is not a photosynthetic pigment. 9, most green plants do not absorb the green spectrum. So 9C. What sugar is produced by Calvin cycle? Glucose is a sugar, right? So it's a carbohydrate, it's a sugar. So it's A. What is the enzyme used in C3 Calvin cycle? It's the Rubisco. Answer is B. What is the benefit of the C4 adaptation? They're doing everything because they are in hot environment to stop the photorespiration, right? Decrease photorespiration. 13. So they're going to have temporal separation, so they can do the carbon dioxide fixation while the stomata are open during the night time, right? So it's the temporal separation. What does CAM plants store? There's no malate given, right? So malate is one possible answer because they're going to store the malate inside their vacuole, malate or malic acid. But because the malate is not there, you can go for water. I said the leaves store water. Right, so the CAM plants are going to store water. CAM plants have a different adaptation to what climate is hot and dry. Why do CAM plants open their stomata at night? To get the atmospheric carbon dioxide for the Calvin cycle. Right, at the end, malate is getting the carbon, so malate can give it to the Rubisco. Next one. In C4 plants, the Calvin cycle takes place in the bundle sheet cells. So 17 is D. 18. What are C3 plants? So C3 plants, 18, plants that do the Calvin cycle in their mesophyll cells. Right, the normal plants. Next one. In C3 photosynthesis, the reactions that require ATP take place in the Calvin cycle only. We put the ATP usage inside the Calvin cycle only, right? So Calvin cycle. CAM plants keep stomata closed in daytime, thus reducing the loss of water. So they can fix into organic acids during the night when the temperatures are cooler. So it's the malic acid, right? So answer is A, 20A. 21, the enzyme responsible for carbon fixation in C3 is the normal Rubisco. The main problem encountered by C3 plants in hot condition is the photorespiration. This leaf structure regulates the opening and closing of the stomata, are the guard cells. Which of these would be considered a C4 pathway plant? It's the sugarcane. So you know the cactus, right? So algae is not like a big plant, it is just a protist. So oak tree, as an example, it's a normal seed tree plant, right? Algae is a protist, it's not even an animal, it's not a plant. 24 is sugarcane. 25, which of these would be considered a camp pathway plant? It's the cactus. The hottest environment is the cactus. 26. On hot dry days, C3 plants will move to the photorespiration. 26 is A. 27. Which part of the leaf is involved in conserving water in dry condition? They're going to close their stomata. So it's D. You will get stomata and stroma for sure, right? So the same, two different choices. So I can just make it this to stoma instead of stomata. So accidentally you might pick stroma, right? So the opening is the stoma, not stroma. 27 is D. Next one, plants like cactus, the succulent, and the pineapple, they are chem photosynthesis, chem 28C. Which type of plants fixes carbon only at night? They are the chem plants. Oxygen becomes attached to RuBP during the C3 photosynthesis. C3 pathway. Last one, all of the following statements are correct, except the wrong one is the first one. These reactions begin soon after sundown and before the sunrise. So I did every single possible question on C3, C4, chem photosynthesis. These are like new parts. You won't find any past exams, right? But there's a little bit more to do, like the factors that going to change the photosynthesis rate, right? And then I can start I think I have Rubisco and cellular respiration and something else, right? Just go through. So what you can do is just write down the C3, C4 chem a couple of times. You will learn all the enzymes and inputs and outputs and everything, right? I think this is not hard, the topic. So if you have downloaded and saved everything, you can leave. I'm done. Bye.
Thank you.